While we've already covered the glial cells, or the supporting cells, it's time to get into the neurons, the actual functional portions of your nervous system. So here, when you start looking at a neuron, you're looking at the structural unit of how the signals are being conducted. You're going to have a cell body, which is this portion right here. Just like any other cell, you've got the cell body that has a lot of components in it. But then what's unique is we have these processes. This one has a whole series of processes coming off, but there might only be one process coming off of an actual neuron. It all depends on what that neuron's purpose or function is. So let's start looking at the neuron and the cell body of the neuron. Now, another term for cell body of the neuron is soma. So sometimes you'll see cell body, sometimes you'll see soma. But the nucleus and organelles are all found in the cell body. But what's unique, there aren't any centrioles. The reason there aren't any centrioles is because the neuron is amitotic. You're not going to have neurons simply dividing to make more neurons. It doesn't work that way. That's why any damage to your nervous system could potentially be a permanent damage. Sometimes it can regenerate and heal, but if there's enough damage, it might not be able to regenerate and heal itself because you simply can't keep making new and new neurons through mitosis. Now, I have listed here as a major biosynthetic center. Well, that's because you're making all of these neurotransmitters. All these neurotransmitters that are being secreted have to be produced somewhere. And then we have these things called Nissl bodies. Usually they're all around in the actual body itself. The Nissl bodies are a unique name, a unique structure to the neuron. But they're basically the same thing as your rough ER. So think about basically production. You're producing here. Neurofibrils. Neurofibrils help to give structure to the neuron. Keep that cell body in the proper shape, keep it open, keep it functional. And the axon hillock. Now, I know we said we're talking about the cell body, but technically the axon hillock is part of the cell body because you haven't quite gotten to the axon yet. The axon hillock is kind of a triangular shaped structure. It starts in the whole body and then thins down until it makes itself into the axon. So the axon hillock is going to be where the axon will begin, but it's still part of the cell body. All right, off the cell body though are all these processes, things like dendrites and axons. We'll get to those in a second. But we have terminology, the term tract. So a tract is going to be these processes coming off in the central nervous system. Now, of course, they had to give different terminology to the peripheral nervous system. Peripheral nervous system, it's called nerves. So when you have these axons coming off, these long axons potentially, if it's in your brain or spinal cord, so in the central nervous system, it's called a tract, a grouping of axons. But if it's out in the peripheral nervous system, these same grouping of axons are now referred to as nerves. So two different names, exact same things, grouping of axons, just depends on where they're located. Let's start with the dendrites. These are all dendrites. You're looking at the things that are coming off of the cell body. Those are all dendrites. Now the dendrites are usually very short. They're not really going to branch out very far because they're not able to have long-range transmission. Their signals are based off of graded potentials, so these short-lived signals. That's why they're always close to the cell body, not too long or far away. They are your input regions. You kind of see this shaded area right here. That's an axon that's coming towards the dendrite. So it's going to send the neurotransmitters across. The dendrites receive those neurotransmitters. The dendrites receive stimuli from your skin or your muscles. So the dendrites are the input going into the cell body. The flip side, the axons, They're carrying information away from the cell body. Axons always carry away from the cell body. Now, axons, they can be very long or very short. All depends on what they need to connect to. But the axon uses the action potential as its mode of transporting a signal. So it starts from the axon hillock. This right here would be the axon hillock. It's the junction between the cell body thinning out into the actual axon. Now the term axoplasm, that's the same thing as cytoplasm. But because we're in the neuron, it has a different term, slightly different composition compared to cytoplasm. But axoplasm is going to be what carries the different particles inside of the axon. We then have the axolemma. The axolemma is the same thing as the cell membrane or the plasma membrane. But again, since it's on the axon, has a unique term to it as far as how the function is going to work a little bit. So axoplasm, cytoplasm, axolemma, plasma membrane or cell membrane. Telodendria. Now when you get down, following this axon way down towards the end, the telodendria are going to be these branches at the end. Not the tips, not the ends, but the branches themselves. Because see how this one single structure branches out into many others? So that is the telodendria, the branching at the end of an axon, telodendria. And then at the very end of those telodendria, those are the axon terminals. Each end here is going to have an axon terminal, known as a synaptic bulb also. They're kind of these little bulb-like projections where the telodendria comes down and it opens up into this bulb-like protrusion. That's where the neurotransmitters will be released when time comes. So we have the axon branching out to telodendria. They're tipped at the very ends with these axon terminals, or these synaptic bulbs. Inside of those end terminals are these synaptic vesicles. The synaptic vesicles are what contain the neurotransmitter. So they're holding and containing this neurotransmitter in this kind of ball-like structure until it's time to release it out of that synaptic terminal. So what are the functions? What do axons do? Well, we already said they are going to generate and transmit action potentials. That's the main purpose. They allow the action potential, the signal, to go from the axon terminal down to the end of the axon. Once you reach the end of the axon, you secrete the neurotransmitter. That neurotransmitter is to go to another neuron, go to a gland, go to an organ, whatever. But it's meant to create a signal in that next step. Note of Ranvier. Now, we already discussed Schwann cells. We've discussed oligodendrocytes. But now, if we are looking at the node of Ranvier, and we're in the peripheral nervous system primarily, it happens a little in the central, but mostly peripheral. Now this happens to show oligodendrocyte with the branches. But a lot of times we'll talk about it in the peripheral nervous system. It's going to be this section in between. Those sections in between the myelin sheathing are going to be the nodes of Ranvier. The reason that's important is all of the voltage-regulated sodium and voltage-regulated potassium channels are compressed into that node because they can't allow for exchange through the myelin sheathing. So the signal kind of jumps to here. A lot of exchange occurs. Signal jumps to the next spot. A lot of ion exchange. Jumps to the next one. It's a very fast conduction this way. And it almost looks like it's jumping gap to gap because the insulation is doing its job. Insulation holds everything in. So it nodes around Ranvier, in between the Schwann cells, and if the oligodendrocytes have some gaps here, as shown in the image, some gaps there. Now I've mentioned axons can be very short or very long. When we're talking about a cell, what is long? you can have some axons running actually all the way from your pelvis region all the way down to your foot. So for one single cell, we're talking microscopic, having that long axon, it's kind of impressive. So these long axons, these are what make up your nerve fibers. So if we're in the peripheral nervous system, that's what makes up your nerves. So when you start looking at how everything's working here though, because if you have this very long axon, you need to get those neurotransmitters and all the structures and everything else down to the end and then back to the cell body. So what's going to happen here is we're going to have what's called axoplasmic flow. Now the axoplasmic flow is the movement of the actual cytoplasm, which is called axoplasm here, inside of the axon. You have what's referred to as the anterograde. So anterograde flow, that's heading down to the axon terminal. So cell body going to the axon terminal. That's anterograde. Retrograde is the opposite direction. It's bringing the recycled components, the waste, the other unused particles, whatever. It's bringing it from the axon terminal back up the cell body. So this axoplasmic flow is going to be either anterograde or retrograde, moving towards the axon terminal or coming back from the axon terminal back up to the cell body. So transporting whatever is necessary up and down the axon. A very unique, specialized flow here. So how do we classify all these neurons? Because we have different functions, different purposes. Well, we're going to have what's referred to as a structural classification and a functional classification. The structural classification is based on how it's built. What is the physical structure of a neuron? While the functional classification is based on what is that neuron doing. We're going to start with the structural classifications. This particular neuron, you'll notice, here's a cell body, has one branch and only one branch coming off the cell body. That means this is called a unipolar neuron. Uni meaning one, so only one process. If you're thinking, hold on, is that an axon, is it a dendrite? It serves as both. You'll notice how that one process branches into two different sides. So you'll have some being dendrite, some being axon equivalent. But they're not really called that because you're looking at one process here. Now, these unipolar are primarily your sensory neurons found throughout your peripheral nervous system. Unipolar. Next up is bipolar. You have two processes coming off the cell body. Now, these bipolar neurons have one axon and one dendrite. One input, one output. Now, these aren't nearly as common as the unipolar or the next one, the multipolar, because they're found primarily only in the special senses. Think eye, ear, things like that. So this is the bipolar. And here's the multipolar. Multi because you can see all the different branches coming off of the cell body. Now this one has what, 1, 2, 3, 4, 5, 6, 7 there. It's could have 10, could have 3, whatever. It's more than 2. Multipolar just means it has 3 or more processes. It'll have the one axon, which is this thing, and all the rest are dendrites. So we have one axon and multiple dendrites at this point. This is most commonly found as a motor neuron. That means motor outputs. So motor neurons a lot of times. While the unipolar was mostly as sensory neurons. Now, while the unipolar and multipolar might be the most common types, the bipolar is there. It's pretty common, but not as common as the other two. There are some other more unique structures. Here, we're going to have these pyramidal cells. You notice they have a whole unique look to them. We then have these Purkinje fibers up from the cortex. Again, a very unique structural look to it. And you even have these olfactory fibers. Again, very unique structural buildup. They're specialized based on what they need to do. So your body has, yes, the common ones, unipolar, bipolar, multipolar, but then also specialized structures that do a very specific task and purpose and their locations. So these are all the structural classifications. The functional classification is based on what is being done. First off, afferent or sensory neurons. We already mentioned this in a previous video. Afferent, sensory, bringing information in. So that means it doesn't matter what the structure is. If this neuron brings information into the central nervous system, it's going to be an afferent or sensory. You then have an efferent or motor. Again, we mentioned that. As long as information is leaving the central nervous system, doesn't matter what the structural classification is, if it's leaving, its functional classification is motor or efferent. And then we have these interneurons. Like the name implies, the interneurons are kind of in between. So you might have a sensory neuron coming up to the brain. Then you have an interneuron before the motor neuron comes back out. The interneuron are making connections. You can see how the interneurons are storing, processing, retrieving information. So you'll have a lot of these interneurons in between the different neural networks of your central nervous system. So these interneurons. So afferent or sensory in, efferent or motor out, or the interneurons kind of play a part in between. So structural and functional classifications of the neurons. Structural based on what they look like. Functional based on what they do. And it's all based on what the neuron is itself. Cell body, dendrites, axons.